So the first example I would like to show you is from adeno-associated virus. So it's a UK-based cell and gene therapy company. They're expressing the adeno-associated virus in Sf9 cells using a baculovirus expression vector. And the objectives in this project was to compare the protein profiles of 6 different batches of drug substances from 2 different AAV products. So, and the question is the purity and protein profiles consistent between the batches? And they also asked us, what is the reproducibility of your method if we are going to send more samples later on for the same analysis? And the second objective, are there any specific wholesale proteins that can be potentially problematic or of concern for product stability or eventually for patient safety? So both compared to what alpha-lyase has seen in other AAV products, but also what is known in the literature from classical biologics. So if we look at some of the data, so here we only look at the viral proteins resulting from the analysis. So what you can see is the total viral content in nanograms per ml for the 6 different batches. So we can see for product 1, the viral content is quite consistent between the 3 batches. For product 2, 2 of the batches were quite similar in the content, whereas the batch number 6 had a much higher content or concentration of the viral proteins. When we looked at how reproducible the method was, we did a triplicate analysis, and you can see in the bottom table that the coefficient of variation is very good. Typically, it's between 3 and 6% here, 7%. Normally, it's within 10%, we say. So this is much higher than what you can obtain, for example, with a standard ELISA assay. Then the second part of the analysis was to look at the wholesale proteins detected in the AAV. So the information is of course confidential for this client product, so that's why it has been discussed for some of the proteins. So, but you can see the principles of the analysis. So you get a list of proteins, you get the amount, you get the theoretical isoelectric point and theoretical molecular weight of each of the protein. You get the names and the database accession number, so you can look up the protein in the original database and learn more about this protein. You can also evaluate it for potential unwanted properties in your product. So what I've just highlighted here are 4 different proteins that we often observe. So one of them is ubiquitin. In itself, it's not problematic for the patient, but ubiquitin can mark proteins for degradation. So it is a potential problem that if large amount of the drug is ubiquitin-related, then it might be partly degraded. So you want to monitor this protein. Also, there is a peptidyl isomerase, and this protein is not known from Sf9 cells to be problematic, but from CHO cells in MAP product, it has been known to cause some product aggregation due to post-translation modifications of the MAP. So this proteins you may also want to monitor and perhaps optimize your process to remove this protein.